Alright, so the ultrastructure of a plant cell. All these structures so far described in animal cells are also found in plant cells, with the exception of centrioles and microvilli. Okay, so centrioles and microvilli are not present in plant cells. Good information. The plant cell structures that are not found in animal cells are the cell wall, the large central vacuole, and chloroplasts. So these are special to plant cells, not present in animal cells. These are all shown clearly in figure 1.27 and 1.28. The structures and functions of cell walls and vacuoles have been described on page number 5. Chloroplasts. Okay, so the structure of the chloroplast as seen with the electron microscope is visible in figure 1.27 to 1.29 and at a higher resolution in figure 13.6. Chloroplasts tend to have elongated shape and a diameter of about 3 to 10 micrometers compared to 1 micrometer diameter for mitochondria. Like mitochondria, they are surrounded by two membranes forming the chloroplast envelope. So chloroplasts are double membraned or is a double membraned organelle. Also like mitochondria, chloroplasts replicate themselves independently of cell division by dividing into two. Okay, nice information. The main function of chloroplasts is to carry out photosynthesis. Photosynthesis is basically the process by which the plants make food for them using sunlight, water, and carbon dioxide. So the food is formed in the form of carbohydrate. Okay, so chloroplasts are an excellent example of structure structures related to its function. So a brief understanding of their structure will help you to understand their function during the first stage of photosynthesis. Okay, so now here photosynthesis, the process of photosynthesis is given very shortly. You will not understand anything from here, but once we go to the chapter of respiration and photosynthesis, we have to learn them in great detail. So yeah, you do not expect to understand anything from here, but in the chapter of photosynthesis and respiration, you will understand everything in great detail. And in your O levels or the previous book, you have learned that photosynthesis is basically a normal equation, short equation. But it is a metabolic process which requires many steps. Okay, so we will see that later, but let's just read it out. Okay, but you will not understand anything from this chapter. During the first stage of photosynthesis, the light-dependent state, light energy is absorbed by the photosynthetic pigment, particularly the green pigment chlorophyll. Some of this energy is used to manufacture ATP from ADP. So in chloroplast, respiration also takes place. Okay, but the main function is photosynthesis. An essential stage in the process is the splitting of water into hydrogen and oxygen. The hydrogen is used as the fuel which is oxidized to provide the energy to make ATP. This process, as in mitochondria, requires electron transport in membranes. This explains why chloroplasts are in a complex system of membranes. The membrane system is tightly organized. It consists of fluid-filled sacs called thylakoids, which spread out like sheet in 3 dimensions. In place, the thylakoid form flat, disc-like structure that stack up like piles of coin, many layers deep, forming structures called grana. From their appearance in the light microscope, grana means grains. These membranes contain the photosynthetic pigment and electron carriers needed for the light-dependent stage of photosynthesis. Both the membranes and whole chloroplast can change their orientation with the cell in order to achieve the maximum amount of light. So that's an adaptation of the plant cells so that maximum efficiency so that the photosynthesis can be carried out maximum efficiently. Okay, so the second stage of photosynthesis, the light independent stage, uses the energy and reducing power generated during the first stage to convert carbon dioxide into sugars. This requires a cycle of enzyme controlled reaction called the Calvin cycle and takes place in the solution in the stroma, the equivalent of the matrix in the mitochondria. The sugars made may be stored in form of starch grains in the stroma. Okay, this is one information. The sugars may be stored in the form of starch grains in the stroma, or may be transported to other areas like fruits where they will be stored. So we recall that plants store glucose as starch, while animals store glucose as glycogen in the muscles. Okay, yeah, so starch is not present in animals and glycogen is not present in plants. The lipid droplets also seen in the stroma as black spheres in electron micrographs are reserves of lipid for making membranes or from the breakdown of mitochondria in the chloroplast. Okay, like mitochondria, chloroplasts have their own protein synthesizing machinery, including 70 Svedberg ribosomes. You know, S is Svedberg, which is the unit used for the measuring of the ribosomes. Oh, you do not need this in this book. And a circular strand of DNA. Okay, in electron micrographs, the ribosomes can be seen as small black dots in the stroma. Fiber of DNA can also sometimes be seen in small clear areas in the stroma. As with the mitochondria, it has been shown that chloroplasts originated as endosymbiotic bacteria. In this case, photosynthetic blue-green bacteria. The endosymbiotic theory discussed great detail on chapter 17. I discussed about endosymbiotic theory, right? So it is basically how two organisms are symbiotically mutually related, and like the chloroplasts and mitochondria, they form endosymbiosis with the cells. Okay, so yeah, I explained this. Nothing much to explain here, and these mechanisms of photosynthesis you learn in great detail and in a better explanation in the later chapters. Okay, so let me show the structure of Thylakoid and Granum. So look at this. This is Thylakoid, and Thylakoid is stacked one upon another to form the structure called Granum. Okay, this structure was not present in the book, so I had to go to the internet and show you this diagram. And look, the stroma is basically the matrix inside the chloroplast. Okay.